Good morning, ma'am. This is Aldrin Dave Diacinto of 33-A1-8. This is my midterm laboratory exercise semen analysis, and the lesson objectives are: explain the importance of semen analysis, analyze the forensic examination of semen. On my presentation, I will discuss also the semen and sperm origin, composition, and nature. The difference between presumptive test and confirmatory test for semen. The steps in collecting evidence with semen. And last, the importance of semen analysis in criminal investigation. Semen and sperm origin, composition, and nature. Semen, the male reproductive fluids containing spermatoza in suspension. Semen from Latin literally means seed. Most of the fluid in semen is made up of secretion from the male reproductive organs. Semen containing proteolytic enzymes, critical acid, acid phosphates, and lipids. Sperm. Sperm is a system of tiny tubes in the testicles. This tube called the seminiferous tubules house the germ cells that hormones including testosterone, the male sex hormones cause to turn into the sperm. It looks like a tadpoles. Fluids contribute by the seminal vesicles are approximately 60% of the total semen volume. These fluids contain fructose, amino acid, citric acid, phosphorus, potassium, and hormones known as prostaglandins. The difference between presumptive test and confirmatory test for semen. Presumptive test for detecting semen is called the acid phosphates or AP test. The AP test detects the enzyme acid phosphates that is secreted from the prostate gland. Confirmatory test is used under UV light. Semen fluoresces, making it visible to investigators to collect samples from a crime scene. RSID semen strip test is one of the examples of the test. These are the steps in collecting DNA evidence with semen. First, absorb the suspect liquid semen on a clean cotton swab. Air dry the swab and pack in clean envelope with sealed corner. Second, submit small and dry suspected semen stained objects to the laboratory. Pack in clean envelope with sealed corners to prevent stain removal. Third, documenting the scene. Fourth, processing evidence at the scene. Last, completing and recording the scene investigation. When investigators find a piece of evidence, they place it in a paper bag or envelope, not in a plastic bag. This is important because plastic bags retain moistures, which can damage DNA. Direct sunlight and warmer condition may also damage DNA. So, officers try to keep biological material at the room temperature. They label the bags in the information about the material is, where it was found, and where it will be transported. These are chain of custody procedures, which ensure the legal integrity of the sample as they move from collection to analysis. The importance of semen analysis in criminal investigation. Semen analysis is an important laboratory test and should be true in the same way in another diagnostic assay is used in determining treatment plans for infertility. Semen analysis is very important in assault cases as it is very useful for detection of semen in clothes, isolation and identification of seminal stains found in the various suspected are such as clothes, bedsheet, pillows, blanket, and etc.